The blood agar test. Blood agar is used to differentiate bacteria based on hemolytic activity. Hemolysis is the process of destroying red blood cells in order to use their contents as a nutrient source. To begin, we will be inoculating 4 different bacteria, 3 knowns and 1 unknown. You will want to divide your plate into quadrants by drawing a cross on the back of the plate. Then you will need to label each quadrant of the plate with the bacteria that will be inoculated. Once you have completed labeling the plate, flip it lid side up. There are 2 different ways to inoculate the blood agar plate. Both will require aseptic technique. For the first technique, it is a simple plate inoculation. Make sure when you are inoculating the plates that you know which quadrant should contain which bacteria, and that they are inoculated in the correct spot. Here, aseptic technique is being used to do a simple straight line inoculation on the plate. The bacteria you will be handling can be very virulent, so be very careful when carrying out inoculations. Be sure to use universal precautions and dispose of all waste in the biohazard container. The inoculation method being shown here will be repeated for each of the quadrants in the plate until all 4 quadrants are correctly inoculated. This is a simple plate inoculation. A straight line technique works very well for observation of results later on. In the second technique, a straight line inoculation is also completed, but is then followed by stabbing the agar with the loop. 4 to 5 small punctures into the agar will introduce bacteria into the media and into anaerobic conditions. Shown here is a completed blood agar plate. Shown here is alpha hemolysis, also referred to as incomplete hemolysis. Alpha hemolysis results in incomplete breakdown of the red blood cells. This often causes a darkening in the agar surrounding the growth of the bacteria, or even a greenish tint, which occurs when iron left behind reacts with oxygen. Alpha hemolytic streptococcus are classified as group B strep using the Lancefield classification system. Now shown is gamma hemolysis. Gamma hemolysis is really just no hemolytic activity at all. In this case, Enococcus faecalis did not produce hemolysins responsible for hemolytic activity. Now shown is beta hemolysis. Beta hemolysis is the complete breakdown of red blood cells. With beta hemolysis, there will be a clearing in the agar surrounding the actual colonies themselves. The clearing is due to diffusion of the hemolysins produced by beta hemolytic activity. Beta hemolytic strep are classified as group A streptococcus by the Lancefield classification system. They also tend to be the most virulent of the streptococci. Blood agar is used extensively in diagnostic laboratories for the identification of hemolytic activity in infection-causing bacteria.